Hello, hello. Welcome, everyone. I'm Kevin Curran with Bridgepoint Bio. And in this video, I will explain the biology of gene editing. How does it work? And I'll do so in the context of Casgevy. It's the first FDA-approved medicine that relies on gene editing. This was approved in December 2023 for patients with sickle cell disease. So here we're modifying red blood cells so that they correctly produce hemoglobin to resolve the disease effects of sickle cell. So first, I'm going to explain this from the patient perspective, and then I'll dive into the genetics. This is an ex vivo therapy, which means that the gene editing tools, they're not delivered into the patient's body, into their bloodstream. Instead, we're removing the cells from the body and then genetically modifying the cells and then infusing them back into the patient's body. So the genetic modification is carried out outside of the body. So specifically, bone marrow stem cells are removed from the body. We then purify for the bone marrow cells, CD34s, and then CRISPR Cas9 is applied. I'll show you what that looks like in a second. This is guide RNA and another RNA molecule for Cas9, which is an enzyme. Those are introduced into the bone marrow stem cells through a process called electroporation. So it's a little blast of electricity, voltage, and that moves them into the cells. The CRISPR-Cas9 tools there will then modify the genetics of those cells to increase hemoglobin levels, and then those modified bone marrow stem cells will be infused back into the patient's body, okay? And the bone marrow stem cells will then develop into healthy red blood cells, which are producing hemoglobin protein. The hemoglobin binds to oxygen, and it does its job. It distributes oxygen around the body. All right. So what does this look like on the cellular level? If we dive in to look at these tools genetically, I'm going to just shrink my video here. So first step is scientists have to decide what gene do they want to modify. In this case, we're trying to boost levels of hemoglobin. So scientists are going to design guide RNA that will specifically bind to a genetic region, which is then going to be modified. It's going to be modified using this Cas9 enzyme. So the enzyme, once Cas9's in the cell, the Cas9 enzyme forms, right? Enzyme is capable of delivering a double-stranded break to DNA. The Cas9 will form a complex with the guide RNA. The guide RNA is going to deliver this enzyme directly to the genetic region where scientists want to make the modification, okay? And like I said, these are cells outside of the body. They're electroporated in. They then will migrate inside the nucleus where they can engage with chromosomes, okay? They've got nuclear localization signals to help them travel into the nucleus. So now we're inside the nucleus of those bone marrow stem cells, right? In a laboratory. This is ex vivo. We're not in the patient's body at this point. And so now, here in gray, you're looking at this is the double-stranded DNA, okay? So this is the genetic region that we're trying to modify. The guide RNA will form complementary base pairing. It will attach to the specific region, and it does so through complementary base pairing and also by identifying and binding to this PAM region, which is another small genetic region. So the two of those will bring this guide RNA into place. There will be an affinity and attraction to that genetic region, and the guide RNA will then, it's attached to the Cas9 enzyme, a nuclease. That's kind of the power center. That's the Cas9 is going to deliver this double-stranded break to that specific region of DNA. So Cas9 creates a double-stranded break. So both sides of the sugar phosphate backbone of helical DNA is chopped open. Okay. Now at this point, you just let natural cell biology processes try to fix that double-stranded break, right? We've evolved those systems, DNA repair mechanisms. Now, the most common path a cell will take when trying to repair a double-stranded break, it's called non-homologous end joining, okay? And let me move my video out of there. NHEJ, this is what's happening with the Casgevy drug for sickle cell. This means that the double-stranded break happens, and now the cell is going to reseal that break, right? Trying to minimize DNA damage. But in the process, there's a disruption, okay? So non-homologous end joining, it's the most common route, but it's also error-prone. So this error-prone piece can be taken advantage of with this therapy because you're actually disrupting that genetic region. You're letting it repair, but with the disruption, then you select for cells with the right disruption. Okay, so that's what's happening with Casgevy. Okay, I'm not going to get into homology-directed repair. That's another route. It happens with lower efficiency, and that's where we're going to try to introduce repair template to actually insert DNA. That hasn't been working as well with CRISPR-Cas9. It's very low efficiency, and that's why there's a lot of interest in base editing and prime editing, because they appear to be better at actually introducing corrected sequence. But right now, CRISPR-Cas9 seems to be very successful at disrupting a genetic region. That's good if you want to knock a gene out, remove its function. And in the case of sickle cell, that gets the job done. So let me just move this over here. This is probably more detail than a lot of folks want, but this is the genetic change that's actually happening with Casgevy. And I need to explain fetal hemoglobin really quick so everyone can appreciate this. But as adults, our red blood cells rely on adult hemoglobin. It's adult hemoglobin protein. It's in a heavy abundance in our red blood cells, and that's what's grabbing oxygen and distributing it to tissues in our body as our blood circulates. Well, it turns out when we're babies in utero, in fetuses, there's another hemoglobin that is working. It's called fetal hemoglobin, right? Makes sense. Part of the normal process of becoming an adult is you turn off that gene. So you shut down fetal hemoglobin protein and you start turning on adult hemoglobin. That's just natural adult development. So what the folks at Vertex Pharmaceuticals and many other groups have been aiming to do here is their strategy is let's go back and prevent the suppression of fetal hemoglobin. So let's remove that down regulation, and so that way fetal hemoglobin turns back on in sickle cell patients. Because if you have sickle cell, your adult hemoglobin has a mutation in it, and so it's not doing the job. It's not helping distribute oxygen. You get sickle cell symptoms. So let's compensate for that by genetically modifying fetal hemoglobin to turn it back on in adults. So that's what's happening with Casgevy. So here, if we look at the genetics here, this is a genetic region called BCL11A, and the Casgevy approach is to disrupt this because this is a regulatory region that controls how fetal hemoglobin operates. So if you can deliver the Cas9 enzyme to break this open. Right here, that's a double-stranded break leading to non-homologous end joining. That leads to indels. Those are insertion deletions. So, errors being inserted into the BCL11A gene. That disrupts it. Therefore, BCL11A can no longer shut down fetal hemoglobin, and so you get elevated levels of HbF, which is fetal hemoglobin. So you're just allowing that to turn back on, sort of prolonging the fetal state of hemoglobin, and that does the job. That compensates for the mutation in sickle cell patients. All right. I hope that was clear. Feel free to leave comments if you want me to kind of do a deeper dive on anything with this topic. All right. Take care.